Hi everybody, we're going to start on the nuclear functions of the cell, and in your course pack this is the middle of page 29 and we'll go through page 30 as well. We're going to talk in this PowerPoint about DNA replication and protein synthesis, both of which are nuclear functions of the cell. Okay, so just some groundwork here. You may wonder why we need to replicate DNA. Replication is just another word for copying DNA. And we're going to look at the cell cycle in just a moment and find that there is a major portion of the cell's life cycle known as interphase. And during a very small portion of interphase, there is a 24-hour period known as S phase. And you might think of S as standing for synthesis. So the S phase of interphase is actually where we copy the DNA. So I'm just going to make a note here that S stands for the synthesis portion. Thinking way back to our earlier chemistry, the synthesis process is also known as dehydration synthesis or anabolism. And in fact, that's what we're going to do. We're going to build an extra copy of our DNA. The reason we're doing this is so that each new cell will have its own identical copy of DNA. You know, we're going to go through mitosis, which eventually is something that makes two cloned cells identical to one another. And of course, that's where we're looking at DNA as being a part of that process. We're also going to look at single-stranded chromosomes that become double-stranded. I'll show you what that looks like, and then we'll talk about how double-stranded chromosomes do get separated. So mitosis is a topic that you'll cover in more detail in lab, but I'm just giving you a piece of that process here in lecture by talking about DNA replication. So first I'd like to show you what a chromosome looks like. When it's time for a cell to undergo replication or copying of its chromosomes, all of the DNA in the nucleus gets in one of these forms here called a chromosome. The chromosome on the far left is what we call a single-stranded chromosome. And the way I know that is because it looks like an airplane propeller. There is a single arm on top up here. We call that a chromatid. And there's also an arm on the bottom called a chromatid as well. This piece in the middle here, it's called a centromere. All right, so in S phase of interphase, keep in mind S phase stands for what? That's synthesis, right? And that's where we copy the DNA. So in effect, what we're doing is we're copying a single-stranded chromosome into a double-stranded chromosome. So this one here in the middle that looks like an X, that is our double-stranded chromosome. It has two identical chromatids on top. They are identical to each other, and these two on the bottom are identical to each other. So the tops and the bottoms are actually different from each other. It's the two arms on the top that we call sister chromatids, and these two arms on the bottom that we call sister chromatids. And again, the entire chromosome is held together by this belt buckle in the middle called a centromere. Now, later on in the cell's life cycle, we undergo mitosis, which is the division of these chromosomes, and we actually separate the sister chromatids into single-stranded chromosomes once again. But, you know, they're going to be identical. These two arms on the top are identical to each other, and these two arms or chromatids on the bottom, they all are also identical to each other. So here again is a look at sister chromatids. On the left, we have unreplicated chromosomes, and on the right, we have replicated chromosomes. And the way I know that is because an unreplicated chromosome has only a single chromatid on the top and the bottom. Replicated chromosomes, like you see on the right-hand side of this picture, have two sister chromatids on the top and two sister chromatids on the bottom. What they mean here by homolog or homologous chromosomes, notice these two chromosomes look absolutely identical. So they're just different colors in this picture. That's because when we think about our total number of chromosomes, each of our body cells has 46 total chromosomes. Each of those is a pair of chromosomes. Let me put it to you this way. Most people have 2 feet. When you buy shoes, you buy a pair of shoes. Those shoes are identical. One, of course, for the left foot, one for the right foot. So if you say that in your closet you have 23 pairs of shoes, we can count and find that you have 46 total shoes, right? one for the left foot, one for the right foot, but you got only 23 pairs. That's how it works in our cells. We have 46 total chromosomes, and we have, in other words, 23 pairs. So when you think of a homolog or homologous chromosome, they are identical pairs of chromosomes. In fact, these could be homologs over here. They just happen to be unreplicated. Okay, so the reason we're talking about this is when you discuss meiosis in Bio 211, we'll come back to this concept and explain that homologous chromosomes separate from their twins. So it'd be like taking all 46 of your shoes, you remember 23 pairs, right? And then separating all the left feet into a pile and all the right feet into a pile. Okay, and then you would give your friend your left shoe and you would keep your right shoe. That's kind of how this would work in meiosis. This is a cell's life cycle. It's called a cell cycle, and this cell cycle we can start anywhere. I'll start here at gap 1. I'll make this my starting point. Gap 1 is sometimes known as growth 1, and it is during this time period that the cell will grow and make new organelles, proteins, and prepare itself for becoming larger. We then travel around to this S phase. This is synthesis. This is where DNA replication occurs. In other words, this is where we copy our DNA. We then travel around to another growth period. It's sometimes called gap 2 or G2 or a growth 2 period. In this period, we're actually preparing for the upcoming event, which is mitosis. You'll notice that mitosis does not include the rest of this cycle. The rest of this cycle is known as interphase. Okay? And only these four processes here, prophase, metaphase, anaphase, and telophase, are actually part of mitosis. You may have heard of IP-MAT before, which is a way to keep this straight, but you must remember that mitosis is only PMAT, okay? And interphase is not part of that replication process. So you'll notice that mitosis here is a very short portion of the life cycle. In fact, it's only about 10% of that cell's life cycle. The other 90% of the cell's life cycle is spent over here in interphase. So once the cell divides in two, it goes back into the gap 1 or G1 phase, travels through S, through G2, and then at some point later it'll undergo mitosis once again. Just as a side note, cells that are cancerous in nature may finish telophase and not go through interphase. Instead, that cell may finish telophase, go right back into prophase, pass through metaphase, anaphase, telophase, go right back to prophase. And so that's how cancer cells sometimes misbehave, because they're not listening to cell signals like they're supposed to be. So when DNA replication occurs, we're going to take this strand of DNA. Remember, it's a double-stranded helix or a double helix, and we're going to separate that DNA strand. The reason we're doing this is to expose those nitrogenous base pairs. So there are enzymes that come along here that help to unwind and untwist this DNA double strand. There is an enzyme called DNA helicase that will untwist this double helix from itself. And then there are enzymes that literally cut open this DNA. Now, if you've got 46 total chromosomes, this replication process occurs in multiple chromosomes in these bubbles that kind of blister up all over our DNA. By doing it this way, we can rapidly increase the speed at which we can complete DNA replication, and the process doesn't have to take that long. So in this picture, you can see that there are some base pairs already being brought in here, and they're binding to this parent strand. This parent strand serves as a template, and the daughter strand is the one that's being built off of this parent strand. Okay. So let's take a moment to look at this double strand of DNA. You'll notice that now we can see these base pairs up close. Just want to remind you of the base pairing rules. Okay, we know that adenine always pairs with what other base? Always with thymine. Likewise, cytosine, or C, always pairs with guanine. Okay, so you can see the parent strand here serves as our template, and if you just work yourself along, you can see G's and C's here paired together. This C should pair with a G, That A should pair with a T. That A should pair with a T, and so on. On the opposite side, let's look at this parent strand. This G here should pair with a C. This T should pair with an A. That A should pair with that base T. So you actually have a built-in self-check. If you look at the opposite parent strand, you'll find that that's all we're doing. We're just simply creating a base pair. Okay, so when you get done with DNA replication, you will eventually have made a double-stranded chromosome. So we're heading up here in this direction, and we're going to finish the replication process. So ultimately what you have are sister chromatids here on the top, and you have sister chromatids here on the bottom, and we will separate those sisters from one another during mitosis. So on your worksheet, what we end up having for each new double-stranded DNA is an old parent strand or template strand and a new daughter strand. Now, at the bottom of your page, on page 29 in your course pack, you'll have an opportunity there to do DNA replication. So the parent strand there is starting off as ATC. So let me just give you a moment to fill in those answers, and then I'll provide the correct ones for you to check by. All right, so the new daughter strand for the parent strand should be TAG, TAC, CGT, AGC, TATG. So easily you can look at the other old parent strand, and that serves as our new daughter strand. I'm hoping this is an easy process for you. Keep in mind there is no uracil here because, well, where is uracil found? Only in RNA. Okay, so don't get these two confused. There's no uracil here when we're doing DNA replication. Now, if you'll follow me over to page 30, we will now talk about protein synthesis. So we're back to the cell cycle again. What is this portion of the cell's life cycle called that does not include mitosis? That portion is called interphase. And of course, mitosis only includes prophase, metaphase, anaphase, and telophase. The process of making proteins occurs here in G1. So we make new proteins and organelles. The cell grows larger during this time period. So we're now going to focus on how we make new proteins for the cell. In order to talk about proteins, let's talk about RNA structure for just a moment. You may recall that RNA is single-stranded as opposed to DNA, which is double-stranded. RNA also has this particular base here called uracil that replaces thymine. So notice, no thymine in this list. We also see ribose, which is a 5-carbon sugar. It's similar to deoxyribose, but it gives the name for RNA. This particular page summarizes the difference. Number of strands, thymine and DNA, uracil and RNA. Let's just circle those. The sugars are different, deoxyribose in DNA, only ribose in RNA. And DNA replication is only going to occur inside of the cell's nucleus, while RNA will occur on ribosomes outside of the cell's nucleus. So let's talk about what you see in this picture. This portion of the cell is called the nucleus. You can of course see the plasma membrane out here around the cell. And of course this stuff out here is called the cytoplasm. And down below here, this thing is a ribosome, which we haven't talked about, but I hope you have heard of that before. It is a small little sac that is found in the cytoplasm of many of our cells. All right, so here's the catch. DNA is housed in the nucleus of the cell. It is not allowed to leave the cell through a nuclear pore. For one, it's too large to exit through the nuclear pore. And for number two, if you left that nucleus with the DNA and it was destroyed in the cytoplasm, you would no longer have a brain for this cell. Your blueprint would be destroyed and you would have no way to find a new blueprint. So that DNA is going to stay inside the nucleus. Instead, we're going to send a copy of that DNA, the portion that is going to help us make a protein, through this opening right here called a nuclear pore. That RNA molecule that travels out here into the cytoplasm, hey, if it's destroyed, you still have the original blueprint, the DNA, inside the nucleus. So, it'd be sad if that RNA was destroyed, but unfortunately, or fortunately, actually, we would have a way to recopy and make another RNA molecule from the DNA blueprint. You're looking at two processes here. Inside the cell's nucleus, we see a first process called transcription, and then out here in the cytoplasm on the ribosome, we see a second process called translation. So let me just kind of give us an outline here so we can look at this. Transcription occurs first, translation occurs second. Transcription occurs in the nucleus, and translation occurs in the cytoplasm on ribosomes. Let me give you a helpful hint. Both words begin with trans. You have a C for transcription, and that's alphabetically before L, right? That helps you keep them in the correct order. Let me give you another second helpful hint. Transcription rewrites DNA into RNA. Okay, let me fix that. All right, so there's an R right there. Transcription has an R because we're making, of course, RNA. Translation takes RNA and builds amino acids. Think way back. What are amino acids the building blocks for? They're the building blocks for proteins. See that A right there? That tells me we're making amino acids. So use these built-in clues to help you keep the basics straight for protein synthesis. All right, so transcription is the first process. Where does it occur? That's in the nucleus. And where does translation occur? That's on ribosomes in the cytoplasm. There are three types of RNA involved here. M stands for messenger RNA. R stands for ribosomal RNA. And T stands for transfer RNA. Okay, so these letters, these prefixes here give us a great clue what we're going to be working with. So step one, we're in the nucleus of the cell, and we're going to take the DNA, and I'm just going to give you a sample strand here. Okay, and we're just going to take this DNA strand and we're going to rewrite it into RNA. Okay, keep in mind we're in the nucleus of the cell and we're going to rewrite DNA into RNA. So T normally pairs with A. A normally pairs with T, but wait a minute, this is DNA transcribing into RNA, so we should put U there. C normally pairs with G, so no change there. C pairs with G, no change. G pairs with C. A normally pairs with T, but hold on. Okay, this is RNA, so we have to replace T with U. T pairs with A pairs with what? U. T pairs with A. C pairs with G. G pairs with C, and A pairs with what? With U. Okay, so in fact, you made a specific type of RNA just now. It's called messenger RNA. Messenger RNA carries codes, and these codes are going to help us build amino acids. By habit, I went ahead and separated this into groups of 3. Each one of these is called a codon, okay? And these codons are going to code for a particular amino acid. So I actually have 1, 2, 3, 4 codons here. Jokingly, I've had students call them condoms before. They're totally not that. These codons code for a specific amino acid. Now, this mRNA strand is going to exit through a nuclear pore. So here's where we are. We're inside the nucleus. I'm drawing nuclear pores. That mRNA strand now exits through a nuclear pore and it heads out here to the cytoplasm to one of these ribosomes. And the ribosomes are where we're going to follow up with translation. Let's talk about translation. I just made an mRNA strand in the previous slide. If I can remember correctly, it was something like this. Okay, so we've got just a set of letters here. I'm just going to use these here because it helps me out. And I'm going to put a ribosome in the background. Okay, remember we're doing translation now, not transcription. And I'm going to use this thing that looks like a clover leaf called a tRNA molecule. So what we're going to do now is perform translation. We're going to use this tRNA molecule to help us read this mRNA strand. T stands for transfer, and of course M stands for messenger. So tRNA reads AUG and helps us translate that into an amino acid. If you have your course pack handy, turn to page 30 and use the codon chart on the bottom of the page. Hey, by the way, please don't memorize the chart. It will always be provided for you. This ribosome, by the way, provides a substrate for us to perform this action. The ribosome has a big subunit on top and a smaller one on the bottom. We call this the ribosomal subunits or rRNA subunits. So I kind of think of the ribosomes as looking like a hamburger bun. So AUG codes for start. Okay. I'm going to give you a moment. Take a look in your codon chart and find CGC. Okay, that one codes for arginine or arginine. Take a moment to use your codon chart and find ACC. Okay, that one codes for threonine. And then now take a moment to find UAA. That one codes for stop. So when I give you one of these on a test, I will always include a start and a stop codon because that tells you you've transcribed and translated properly. Now all we need to do is link these together with peptide bonds, and now we have a primary structure of protein, that bead on a string appearance, and then of course from there we can go to secondary, tertiary, and if necessary, quaternary level structure. So these bonds here are, of course, called those peptide bonds. So just to review, mRNA carries the message. It is a set of codons or groups of 3 base pairs. These codons are going to code for a specific amino acid, as you can see below. Ribosomes perform a site for the translation process. You can see the big subunit on the top and a smaller one on the bottom. These subunits provide a substrate or a site for the translation process. Keep in mind, ribosomes may be free in the cytoplasm or bound to a rough ER molecule. Here you can see tRNA. It stands for transfer RNA. It reads the codon and brings in the appropriate amino acid to the growing polypeptide strand. It always looks somewhat like an upside down clover leaf. Like I mentioned, you'll never need to memorize this codon chart. It'll always be provided for you. They come in different forms and fashions. The one in your course pack may be a little easier to read than this one, but never feel like you need to memorize this codon chart. Okay, so on this particular problem, we're going to do 2 steps. We're going to transcribe the DNA first from DNA into RNA. And then the second step we're going to take is to translate that RNA into amino acids. To be specific, we are dealing with a type of RNA called mRNA or messenger RNA. Now, to do transcription, we simply have to remember that we no longer have T. See, A normally pairs with T, but instead we have uracil. Okay, C's and G's pair. Of course, T pairs with A. G's and C's pair. We have to be cautious though because uracil replaces thymine. All right, so what I recommend doing at this point is dividing this strand of mRNA up into codons. Recall that codons are groups of 3. Okay, and so 3 is what we're going to do here. These are called triplets or base pairs that make up these codons or codes. Now, using the codon chart in your course pack on page 30, let's start with AUG. Okay, on your codon chart, AUG codes for start. That means to the tRNA molecule, they should start translating at this point. Next, let's take a look at CCA. That one looks like proline. The next one is AAA. That one looks like lysine. GCA is our next one. That looks like alanine. And then finally, UAA is one of 3 stop codons. And that tells the tRNA molecule stop making the protein here at this point. So this is a very simple protein chain. It is held together by peptide bonds. So what in fact we have made here is a very simple protein. Keep in mind that amino acids are the building blocks for the protein. So if you are asked to transcribe, your job is to take the DNA, rewrite it as the RNA. If you're asked to translate, you're going to be given a codon chart, and your job is to take the mRNA that you've made and now turn it into amino acids using that codon chart. So when you take a look here at this example, this follows exactly what we've just done. Next, here's another example for you to try. I'm going to pause here. And when you're ready, you can go ahead and proceed to the next slide, which has the answers written out for you. Okay, so I would practice these in order to do well on the test. There's going to be a couple of problems for transcription and translation.